Dr. Hugh is not able to come to the meeting due to a last-minute emergency at work, so I'm going to present on his behalf. So in this presentation, we're going to switch a little bit. This morning, Dr. Mitchell and just now, Dr. Lowe talked about injectable myocell nanoparticles. I'm going to talk a little bit about the implant. So during the next 20 minutes, we're going to review a little bit about disease state and cost, and also the current standard of care. And we will discuss a little bit about, Lucentis, Eylea, this type of biologics in ophthalmology, and some of the drug delivery systems currently on market or under development, and some of the new concepts or new technologies. Population aging, as you all know, is a huge problem. It has profound implications to many aspects of human lives. And it's 50 years ago, in 1950, there's according to a report from the United Nations, they have this aging index, which is defined as every 100 children aged under 15, compared to the number of people who is aged 65 and above. So in 50s, that's around 20%, around 24%, and currently in 2010, it's around 35%. And within the next 40, 50 years, it's going to increase to threefold, to 100%, which means that every children under 15 years old will have one senior that is 65 years and old. You can see the huge challenge we're facing in terms of aging. And in the developed world, the black curve is actually an average of the global average, but in the developed country like U.S., that the problem is even more significant. So this means that most of the ocular diseases are associated with aging, and you all know the cost of the ocular diseases. Globally, there's 40 million blind, 110 million visually impaired, and the cost, to loss of productivity, for blind is $19 billion, and for the visual impairments, that's $23 billion. These are actually conservative numbers. And the major diseases, including AMD, age-related macular degeneration, glaucoma, diabetic retinopathy, and cataract. These are all the diseases, major diseases that impact people's quality of life. And I just take one example using the wet AMD, neovascular AMD as an example, just talk a little bit about the current status. So wet AMD in US, there's 1.7 million patients. Globally, there's 3 million. And every year, there's 200,000 new patients in US and 600,000 globally. And currently, the standard treatment is Lucentis from Biogenetech and Eylea by Regeneron. You can see this number. Their sales revenue is like $1 billion. These are billion-dollar drugs. And this year is predicted to the market for AMD market is predicted to be $4 to $5 billion. And with this Lucentis and Eylea, they are very effective. Patients like it. And so what are the issues? The issue is injection frequency. So these medicines has to be injected every month, monthly. And that's a huge issue because the monthly injection is going to increase the lifelong risk of endophthalmitis, cataract, retinal detachment. Whenever you give injection to a patient's eye, it's going to cause these problems. And also, some people are scared of injection to eyeballs, so it causes non-compliant. And another issue is reimbursement rate. because this type of medicine is very, very effective, so the prescription volume is increasing exponentially. However, we only have fixed amount of money in our healthcare system, so the reimbursement rate is, as you can see from the red curve, it's decreasing also significantly. So, key issue is these are very good medicines, very good therapeutics. The key issue is the frequency of injection. How do we manage that? How do we deal with it? If we look at some basic properties for Lucentis and Eylea, so for Lucentis, the molecular weight is 48 kilodalton, and its VEGF binding constant is around 140 picomolar. And it's halfway, it's around 3 days, 4 days in rabbit, and in human it's around 9 days. And for Eylea, the binding, the molecular weight is higher, so it's more than double, it's 110 kD. And the binding, which means the effective concentration is much lower, it's 100-fold lower. And the half-life is comparable to Lucentis. So let's do a little bit of modeling and just see how these three factors, the molecular weight, the half-life, and the effective concentration affect the duration of treatment. If you can see for Lucentis, this is a case for Lucentis. So you have effective treatment concentration around here, and so that's corresponding to a duration of 30 days. And if we overlay the case for Eylea, since Eylea has a little bit long virtual half-life, and its binding constant, its binding affinity is significantly lower than Lucentis. It's 1 order of magnitude lower. So that make it, the duration of treatment is much longer than 30 days. And so these are the, half-life and their effective concentration. These are two major parameters. And there's another one we can play to increase the duration. That is, you give more drugs, give a larger dose. But that's limited. As you can see with this red curve on the top, you increase the dose, you double the dose, but the duration doesn't really increase too much. So, which leaves us with limited options, which means that you can play with, you can have increase the half-life for the proteins, for the antibodies. But this slide shows that there's a limit to do that. This is a plot put together by Devin Welty, who was our head of a PK department. Based on many years of our internal PK data, he put together this plot. The x-axis is the molecular weight, and the y-axis is the virtual half-life. As you can see, with the increase of a molecular weight, and the half-life increase. However, when you increase molecular weight to a certain extent, the half-life increase get plateaued. So, which means that you can only do so much to increase the half-life in order to enhance the therapeutic duration. Now, what we can do. This is probably a challenge for people in this room and in this field, in drug delivery field. So one of the things we can think about is that we develop controlled, sustained release system. So currently for small molecules, there's quite few on market. So one of them, as you all know, Retisert, it has it's a 3-year non-degradable implant for fluocinolone, and also Ruiwen, and Allergan has Ozdex, that's a degradable. However, for protein, so my apologies, this is a very busy slide, and you probably won't be able to read it. But the bottom line for this one is, these are all the protein delivery systems that's being tested or being developed. There's microspheres, there's implant, and there's depot, there's liposomes. The players involved including Genentech, Allergan, a lot of academic labs. The only product that was on market was neutropene, which is a microsphere formulation, but it was pulled out of the market because of some issues with manufacturing. So bottom line is there's no commercial product for biologics for controlled release system on market. However, there's a tremendous amount of resource in industry as well as in academic put into the development of a controlled release system for biologics. I'm gonna briefly review 6 types of technologies. One is PLGA or biodegradable type of. This is one of a case that where it says lysosome, which is a large molecules with like 14 kD of molecular weight. When you directly put into PLGA, you get drug released, but the dark blue curve says that there's no activity. The released one is not active. Basically, in PLGA, you destroy the activity of the molecules. However, there's ways. We have internal data shows that if we formulate it properly, using proper excipient, we can get released and it's active. So, which means that formulation, we can't do something in formulation to maintain the activity while the drug is released. And another technology is developed by Neurotech. This is an encapsulated cell technology. It's a capsule with a polysulfone materials, and it's a semi-permeable the membrane. And this membrane, inside the membrane, there's a live cells encapsulated in there. So the membrane only allows oxygen and the nutrient to pass into the capsule to feed the cells, so that the cell can secrete CNTF factors. And the factor secreted factor can diffuse out and to fight the disease in retina. And it's currently testing in phase 1/2 clinical trial. And they also are developing VEGF, anti-VEGF using same platform. It's also cell-based inside this cell, and it releases VEGF antagonist to neutralize the VEGF in the vitreous. And the third technology is developed by on-demand therapeutics. This is an implant in which you see the different colors that represent different reservoirs of drugs. You can put different drugs or same drugs in different chambers, and these chambers are all sealed. They can keep the water out and keep dry solid in there, so that if you put protein there, it's going to stay stable. However, when you need the drug to be released, you shine a laser, just a regular laser for ophthalmologist, and you shine laser through a slit lamp and put the laser spot onto one of these reservoir, and then you will drill a hole, and the therapeutic protein is going to release out of there. So this has a potential to provide approximately a year of you can put a year of medications in there, and they can potentially control the release for a year, although it's in a very early stage. And this is another technology. It's a device developed by Replenish and the company. Basically, it is a reservoir device, but you can refill medicine into when the drug is replenished. And on the corner here, it is another type of device. It's also a refill device developed by Foresight Vision, Foresight Laboratory. This is their Foresight Vision 4 system. And also, there are 2 major players in the porous silica system. One is Savida, their tester technology, and another one is UCSD, Dr. Michael Saylor's lab. And they developed this microporous silica where you can load your therapeutic protein or anti-VEGF into this micropores, and by charge interaction, And then these pores will hold the protein and slow release. In this year's ARVO, Cyvita presented that they can release bevacizumab for 3 months. At least that's what they claim they can do. And one more type, this is pagylation. You all familiar with pagylation, which is basically you conjugate your protein onto PAG. But the whole conjugate is soluble in water. But there's a second different type of PAG technology where they cross-link the PAG, and your drug is conjugated to the cross-linked PAG, so that the release is by degradation of the the bond, chemical bond between the PAG. So that using that technology, they can control the release. Some company claims that they can control the release for over 3 months. Although these are all at a very early stage, you all know that this type of conjugation could potentially have problem because it's a new chemical entity. FDA regulation, tolerability. Those are all unanswered questions. This basically is summarize my my talk. So basically, we think that long-acting intravitreal implant system can provide a best option for effectively treating the retinal disease or posterior segment diseases. And non-degradable implant are able to provide a sustained release with low burst over a longer period compared to biodegradable. Biodegradable, you eliminate the needs for removal to remove the implant, but the duration might be shorter. So advances in new ocular drug delivery technologies may fulfill the clinical promise for the future. And this concludes my presentation, and thank you for your attention.
Questions?
So you think that the non-degradable implants, which the Osdex, and there are several other implants available, is the future for the macromolecules also for proteins, because they are using only small molecules?
Yes, that's what's current on market. But for the on-demand therapeutics, that it's a similar concept as Redisert. That Redisert is a silicon cup with a fluocinolone tablet in there with a release port, right? In their technology, they use laser drill hole. I mean, it is for this non-degradable. The good thing is that they can prevent degradation, potentially prevent the degradation or loss of activity of biologics. However, their dimension, the size of the implant, it could be bigger. For the degradable, like Ozdex, it's injectable. You can inject through a 22-gauge needle. For Iluvien, you can inject through a 25-gauge needle. But for Redisert, you have to make an incision, surgical incision, and put it in there. And I believe for on-demand therapeutic, their device, you have to probably also make an incision as well. So there's pros and cons. No perfect system yet. Yeah.
Yes.
My question is on the lysozyme release you showed from PNG and TCL plants.
Yeah. That's an in vitro release study. Yeah.
You said there is degradation of released lysozyme.
Mm-hmm.
What's the possible mechanism? Is it simple aggregation because of too much concentration, or it's because of the byproducts of polymer degradation?
Our understanding is probably acidic degradants generated by the polymer. So by PRG, yeah.
And in the next graph, you showed that it's possible to have 100% active.
Yeah.
If you could share, what was the idea?
We include the excipient there. I couldn't disclose what exactly excipient, but the point is using formulation approach within a novel excipient, you are potentially able to solve the loss of activity issue.
Right. All right. Thank
Okay.
you.
Thank you.
Like, quick question.
I have a question that what concept is working for the diffusion of the molecule for continuously for 3 months like that? What kind, like osmotic pressure, or what kind of force is working for continuously to release the drug?
Yeah, most of the systems are passive diffusion. But osmotic is another concept people are exploring. So using osmotic pressure to drive the reservoir, drive the
So
therapeutic side.
in intravitreal, if you are putting the and then that is releasing, how you are sure that that will be continuously releasing with the same flow rate, diffusion rate?
Yeah, yes. You mean how do you
Right.
make sure it's constantly released, right? Yeah. So you can use osmotic as one of the driving force, and also you can because for the degradable systems, once the degradation kicks in, there will once the degradation kicks in, you will have the increase of release rate. However, there's two directions. Because you have less drug left, so if no degradation, you will have slower release. However, due to the degradation, the release is going to increase. So the two trend merge, and they could get sustained release. Yeah.
Thank you.